Hello everyone, welcome to Botany optional channel for UPSC examination. In this video, we are going to see the development of female gametophyte. In the last lecture, we have seen the structure of ovule, and now in this video lecture, we are going to see how the female gametophyte develop inside the ovule. Alright, this topic has been given in your UPSC main syllabus in a paper number 1 and chapter number 3. Alright. So let's get started. Now, to remind something, we have discussed this structure in which we have discussed this embryo sac. Now, this embryo sac is also called as female gametophyte. And now, in this lecture, we are going to discuss the development of this female gametophyte. That is how this structure, which is called the female gametophyte, develops. That is we are going to see in this video lecture. Now, first of all, you know that this structure which creates the female gametophyte, that structure is called as nucellus. This part of the ovule is called nucellus, and inside this nucellus, the female gametophyte develops. As you can see in this picture, that this embryo sac or female gametophyte is developed inside the nucellus. So, nucellus plays a very important part in the development of the female gametophyte. Alright. So all the things started in the nucellus. That is the process of formation of female gametophyte started in nucellus. Now, if we taken out this nucellus structure only, it will seem like this. Now, if you consider this is a typical nucellus made up of the parenchymatous cells, and there is nothing, totally blank space present in the nucellus. Now, what happens in the first step? The diploid cell of the micropylar end, that is this end, right? Now, you see, you can observe in this is the micropylar end, and this is an chalizoid end. Now, a diploid cell, that is, which is having twice a number of chromosomes, a diploid cell at the micropylar end is differentiated to form the structure called archesporium. Now, in the second step, you can clearly observe here the formation of archesporium. This structure, this first structure, is called as archesporium. This archesporium is developed from the diploid cell of a nucellus. Now, this cell being diploid and also called as the archesporium. Again, this cell also called as the megaspore mother cell, that is MMC. The cell which is diploid in nature, which is developed at the micropylar end of an nucellus of an ovule, it's called archesporium or megaspore mother cell. Now, this megaspore mother cell, which is diploid in nature, undergoes the process of meiosis to form the tetrad of 4 haploid cells. That is, you can observe in this next step, the 4 cells, that is tetrad, are formed by the meiosis of one cell of archesporium, right? All these 4 cells are the haploid cells, that is, they contain only the n number of chromosomes. They do not have the twice n number of chromosomes, but they only have the 1n number of chromosomes. Now, this tetrad, which is formed by the cell of archesporium, are all called as the haploid megaspores. Alright. So the haploid megaspores are developed from the archesporium. They are tetrad, that is, they are 4 in number. Now, this process of formation of tetrad or say haploid megaspore from a single cell, this process is known as the Megasporogenesis. You should carefully remember this. This is a very important step in which the haploid megaspores are formed from the diploid cell through the process of meiosis. This process is called as the megasporogenesis. Now, generally what happens? The megaspore which is present at the chalizoid end, that is, this megaspore which is present at the chalizoid end, is remain functional, while the 3 which are present at the micropylar end, these 3 got degenerated, and only the one which is present at the chalizoid end is functional. That is, out of this tetrad, the 3 cells which are present at the micropylar end in the process got degenerated, while only the one which is present towards the chalizoid end remains the functional. Now you can consider this as the first cell of female gametophyte. This fertile cell can be called as the first cell of female gametophyte. Alright. Now after the degeneration of these three cells, there is only one cell remain which is functional, and it is called as the first cell of female gametophyte. Now we have to see what happens with this first cell of the female gametophyte. Now let us consider this is an first cell of female gametophyte. Now this cell, which has the nucleus and the cytoplasm only. Now this first cell of the female gametophyte undergoes the three successive mitotic divisions. That is, you can see here, this is the first step of mitotic division. This is the second mitotic division, and this is the third mitotic division. Now in the first mitotic division, it results in the formation of two nuclei, and in the second mitotic division, the two nuclei got converted into the four nuclei, as you can see in this diagram. Then in the third mitotic division, these four nuclei undergo the division to form the 8 nuclei. Now, inside the nucellus, we have the 8 nuclei here, right? So, at the end of the third mitotic division, we found the 8 nucleated structure. Now, this 8 nucleated structure again goes through certain changes. That is, one nuclei from each pole come down to the center. That is, one nuclei from the chaliceal end and one nuclei from the micropylar end come to the center, and they act as a polar nuclei. These are called as the polar nuclei. And these nuclei are came from one from the chaliceal end and other from the micropylar end. So in the previous step, we have 4 nucleus at the chaliceal end and 4 nucleus at the micropylar end. Now we have 3 nucleus at the chaliceal end, while the 3 nucleus at the micropylar end, and 2 are present in the center, which are act as a polar nuclei. Now this step is followed by the cellular organization. In this step, the three-celled egg apparatus is formed at the micropylar end. This is a micropylar end, this is a chaliceal end. This end, the three-celled egg apparatus is formed. That is, three-celled egg apparatus is formed at the micropylar end, while the three antipodal cells are concentrated at the chaliceal end. That is, three antipodal cells are present at the chaliceal end, while the three-celled egg apparatus is present at the micropylar end, while two polar nuclei remain at the center. Alright, so this is how the end structure is created. This 8 nucleated structure is typically called as the female gametophyte. Alright, so this typical structure in which we have the egg apparatus, three-celled egg apparatus, then two polar nuclei and three antipodal cells is famously called as the female gametophyte. Now, this female gametophyte is formed inside the functional megaspore, and that is why such a type of development is always called as the endosporic development. That is, the whole female gametophyte is developed inside the functional megaspore, and that is why such a type of development is called as the endosporic development. And as we have observed that only the one functional megaspore take part in the formation of total female gametophyte. That is why it is also called as the monosporic. Alright, so you have to remember this, why it is called monosporic, because only the one cell is got converted into the female gametophyte. That is, one megaspore takes part in the formation of whole female gametophyte. Now there are some exceptional cases as well in which more than one megaspore takes part to form the female gametophyte. In such a cases, such a type of female gametophyte may be called as the bisporic or tetrasporic. But in this case, there is only one megaspore take part in the formation of female gametophyte, and that is why we called it as a monosporic. Alright. So this is how step by step the female gametophyte is created inside the ovule or say inside the nucellus, right? Now let's come back to the first diagram in which we have seen the development of, we have seen the complete developed female gametophyte. Now see, again this is an ovule structure, right? This is ovule, and inside this ovule we have here the female gametophyte. Now this female gametophyte is surrounded by the nucellus. As you must remember that the first diploid cell from which all this started is come from the nucellus. The first diploid cell is divided to form the tetrad of a haploid megaspore, and later out of the four haploid megaspores, three got degenerated and one remains functional. After this, the one functional megaspore, then the nucleus of functional megaspores undergoes the three consecutive mitotic divisions in which it forms the eight-celled nuclei structure. After that, out of this eight, the two come one from each end, that is one from the chalazial end, another from the macropolar end, come at the center, which forms the polar nuclei. And later, after the cellular reorganization, the antipodal cells are set at the chalazial end, while the three-celled egg apparatus is settled at the micropolar end, while the two cells of the polar nuclei remains at the center. And this whole structure is called as the female gametophyte. As the whole female gametophyte is created from the one functional megaspore, that is why such a development is called as the monosporic development. All right. So this is how the female gametophyte is developed inside the ovule. Now I would like to request you to join the Telegram channel of the same name, that is the Botany optional for UPSC examination, so that you will get some extra study material at that platform as well. All right. So thank you very much for watching this video. Please like the video. Please share this video with your friends who are studying the botany subject, and please subscribe to the Botany optional channel for UPSC examination. Again, thank you very much for watching this video. See you in the next one.